Modeling the central dogma of molecular biology. DNA replication. Before we begin, feel free to pause the video anytime to read the words on the screen. This is DNA's double helix structure. It looks sort of like a twisty ladder. The rungs in this ladder are made up of nucleotides. Nucleotides are molecules made up of three major parts: a phosphate group, a 5-carbon sugar, and a nitrogenous base. The nitrogenous bases pair up in complementary pairs and are connected by hydrogen bonds. This is what forms the rungs in the twisty ladder. However, in order for these hydrogen bonds to work, DNA has to have an antiparallel structure. This means that its two strands are going in opposite directions. Most strands of DNA have a 3' and a 5' end. New DNA can only be synthesized in the 3' direction. This is an important concept in DNA synthesis. In order to create new copies of itself, DNA must first be unwound and unzipped. This is done by an enzyme called helicase, which unwinds the DNA and unzips it by breaking the hydrogen bonds between its nucleotides. This creates a replication fork where DNA replication takes place. RNA primer arrives to initiate a new strand of DNA. This is because DNA polymerase, which does most of replication, cannot start a new strand by itself. It can only add on. So an RNA primer comes along and starts a new strand for it. However, remember that DNA is antiparallel. Its two strands have 3' and 5' ends facing in opposite directions, and DNA replication only occurs in the direction of the 3' end. For one strand, this isn't an issue. The replication is happening in the direction of its 3' end. This is called the leading strand. However, the other strand has replication going in the opposite direction, towards its 5' end. This strand is called the lagging strand. For the leading strand, replication is simple. A single RNA primer is necessary. Then DNA polymerase takes over. DNA polymerase is an enzyme that assembles free nucleotides into a new strand of DNA. In the leading strand, DNA polymerase will create a single unbroken strand until the end of replication. Note that all the nucleotides in the new strand are the complements of the nucleotides in the parent strand. While this is happening, an enzyme removes the RNA primer and replaces it with nucleotides. The end result is a new strand of DNA. The lagging strand, however, replicates in a more complicated way. Remember that the lagging strand is facing in the opposite direction of the leading strand, so that the DNA replication is occurring in the direction of the lagging strand's 5' end, but DNA can't be created in the direction of the 5' end of the strand, so it has to go backwards. But how? The answer is that DNA primase creates multiple RNA primers. Then polymerase uses these primers to build several small strands of new DNA. These are called Okazaki fragments. Here we see a second Okazaki fragment being built. Eventually, the Okazaki fragments are connected by an enzyme called DNA ligase, forming a full strand of new DNA. Now there are 4 strands of DNA, 2 new and 2 old. These strands coil up into 2 separate double helixes. One old strand coils with one new strand, and another old strand coils with another new strand. This is why DNA replication is called semi-conservative, because half of the result is old and half is new. Protein synthesis. RNA transcription. RNA transcription is a bit similar to DNA replication in that DNA is used as a template to copy something, except in this case the something is not more DNA, but RNA. But what is RNA and how is it different from DNA? Well, DNA contains instructions for the function of living organisms. Specifically, DNA contains instructions for making proteins. RNA is the one that copies those instructions and then carries them out. So think of DNA as a teacher who has instructions for a project. RNA is a student that takes a copy of those instructions and then actually does the project. That's a major difference. If you want to read the rest, you can pause the video here. There are 3 different types of RNA. Messenger RNA is RNA that actually copies the code from DNA. Ribosomal RNA is RNA that forms ribosomes, which will later play an important part in protein translation. Transfer RNA is also involved with protein translation. It carries amino acids to the ribosomes to form a chain. This image shows a relationship between the 3 types of RNA. The process known as RNA transcription is used to copy DNA to messenger RNA, or mRNA. There are 3 major parts of transcription: initiation, elongation, and termination. We'll start, of course, with initiation. DNA is double-stranded, with one strand known as the coding strand and the other as a non-coding strand. Genetic information is carried on only the coding strand. The coding strand has codons, while the non-coding strand contains anticodons. The non-coding strand is complementary to the coding strand, and is also known as a template strand, since it serves as a template for the RNA being transcribed. Before RNA transcription can begin, DNA has to be untwisted and unzipped. A DNA promoter is a region of DNA that initiates the transcription process. So during the initiation portion of transcription, various proteins called transcription factors attach to the DNA promoter. Only after this does the enzyme RNA polymerase attach to the DNA. RNA polymerase then unwinds and unzips the DNA strand and uses free nucleotides to create new mRNA. Once it's finished, the mRNA breaks away from the transcription complex. But for now, it remains in the nucleus of the cell. Notice that the nucleotides that make up mRNA are not the same nucleotides that make up DNA. mRNA contains uracil, which is in place of thymine in DNA. Like thymine, uracil forms a complementary pair with adenine. Anyway, directly after transcription, the mRNA is in a form known as pre-mRNA. At this point, a 5' cap is added to its 5' end, and a poly-A tail to its 3' end. Also at this point, introns are spliced out of the mRNA, while exons are kept. But what are introns and exons? Essentially, introns are information that the RNA doesn't need to code for protein. Exons, on the other hand, are necessary. So introns are removed and recycled, while exons are kept. The pre-mRNA is now mature mRNA. The mature mRNA is now ready to exit the nucleus through nuclear pore and enter the cytoplasm. This is where translation will take place. RNA translation to protein. The nucleotides in mRNA form a string of letters. An example of a string would be AUGUCAUCGGCAUCA. But how do these letters code for anything? First of all, genetic code is read 3 letters at a time. Each triplet is called a codon. One codon would be AUG. Another codon would be UCA. Each codon codes for one amino acid. This is a dictionary that shows how amino acids correspond to codons. So AUG, UCA, UCG, GCA, UUG would code for methionine, serine, serine, alanine, leucine. Notice how 2 different codons can still code for the same amino acid. But how do these codons get translated into amino acids? Part of the answer is transfer RNA. It's essentially a delivery system. Look at its structure. It has one end that attaches to an amino acid and the other end that contains an anticodon, which is a complement of a certain codon. The tRNA and the mRNA meet up in the ribosome, which is made up of proteins and ribosomal RNA. A ribosome has 2 units, a large subunit and a small subunit, which snap together to process mRNA. Inside the ribosome, there are 3 sites for binding, P, A, and E. The first tRNA binds to the P site, the second enters at the A site. The amino acids bond through a carboxylamine bond. Then the first tRNA moves to the E site, the second moves to P, and a new tRNA enters at A. This happens till a long chain of amino acids has formed. The E site will not be shown in this animation. This is a ribosome. It has a large subunit and a small subunit. It's made up of proteins and ribosomal RNA. The 2 subunits of the ribosome snap around a strand of mRNA in order to begin translation. By the way, this is a codon. It's made up of 3 nucleotides. This is a transfer RNA, abbreviated tRNA. It has an amino acid on one end and an anticodon on the other. These are the P and the A sites inside the ribosome. The transfer RNA will attach to these sites during translation. Now let's observe the initiation of RNA translation to protein. Notice the first codon on this strand of mRNA. It's AUG, and it codes for methionine. This is the start codon. When the corresponding initiator tRNA attaches to this codon, translation will be officially initiated. Elongation begins when a second tRNA, carrying a second amino acid, enters the A site. The amino acid on the first tRNA bonds with the amino acid on the second tRNA. Then the first tRNA exits, the second moves to the P site, and a third moves on to the A site, and so on, etc. But how do these amino acids bond together to create this chain? Turns out it's a peptide bond. Look at this amino acid as it appears on the screen. It has an amine group, and it has a carboxyl group. A peptide bond occurs when the amine group of one amino acid comes in contact with a carboxyl group of another amino acid. The reaction forms a bond and releases one molecule of water, which is why it's called dehydration synthesis. Now let's observe termination of translation. This is a stop codon. It doesn't code for any amino acid. This is a release factor. It recognizes the stop codon in the mRNA. Once a stop codon reads a release factor, translation is terminated. The polypeptide is released and exits into the cytoplasm. Later, the ribosomal subunits break away from the mRNA. What are left was a completed polypeptide chain. Mutations. What causes mutations? Various things such as radiation, chemicals, or natural errors in replication or cellular division. These mutations insert, delete, or move nucleotides in DNA. There are various types of mutations with various effects. The main thing, of course, is how the mutation affects the string of amino acids that will be created. The simplest type of mutation is a point mutation, where just one nucleotide is changed. Look at this string of codons, for example. They code for 4 amino acids. Now, what if a point mutation happened, and the last letter in that second codon was changed from a U to an A? This would make it code for a different amino acid entirely. Now it's glutamate instead of aspartate. But how does this change the resulting polypeptide? Well, if glutamate has similar properties to aspartate, then the single switch in amino acid shouldn't affect the entire polypeptide too drastically. However, if they are different, the polypeptide could easily fold incorrectly and thus become a non-functional protein. In this case, we're lucky because glutamate and aspartate are actually very similar. The polypeptide shouldn't turn out very differently from what it originally was. This type of point mutation, where a new amino acid is coded, is called a missense mutation. There are two types of missense mutations, conservative and non-conservative. You can see that this mutation was conservative. So what effect does this actually have on the polypeptide? Let's take a look at protein folding. Protein folding is a kind of complicated thing. First, there's primary structure, which involves a chain of amino acids. There's also secondary, tertiary, and quaternary structure, but we don't really care so much about those because we're not really focusing on them. This is because primary structure is really affected by the mutation in a polypeptide. A mutation could lead to an incorrect amino acid sequence, which could lead to an incorrect protein folding, which would mess up the primary structure. This is how a mutation can mess up a polypeptide. As a result, the protein as a whole might not be able to function. Let's look at a second point mutation. The first nucleotide on this second codon is changed from an A to a U. This changes the codon from coding for lysine to a stop codon. How does this affect the resulting polypeptide? Well, since coding stops in the middle, the polypeptide is almost certainly non-functional. That's bad. This type of point mutation is called a nonsense mutation, where earlier codons are mutated into stop codons, which makes no sense. This mutation can lead to a defunct polypeptide that's too short. Here's another point mutation. The last letter in the second codon here is changed from an A to a G. What horrible things will this do to the polypeptide? As it turns out, not much. Remember how different codons can code for the same amino acid? Well, in this case, the mutated codon is still coding for lysine just like the original codon. So this mutation is not actually a bad mutation to have. It's called a silent mutation because it actually has no effect on the final polypeptide. There are other types of mutations as well. Look at these nucleotides. Something might happen to make the 6 nucleotides in the middle reverse direction. Clearly, since different amino acids are being coded for, this can negatively affect the resulting polypeptide. This type of mutation is called an inversion mutation and can affect multiple amino acids as well as the order of amino acids. Another type of mutation happens when a single nucleotide is inserted into the sequence. This insertion shifts all the nucleotides after it one space to the right. This would drastically alter the resulting polypeptide, since an entirely new sequence of amino acids would be coded after the frame shift. The polypeptide probably wouldn't function. This type of mutation is called frame shift, and it's really very unpleasant. But how do these mutations actually affect the individual who has them? And what about the individual's offspring? Well, first of all, mutations have many unpleasant effects, such as cancer or genetic diseases. But the question of whether or not an individual can pass a mutation down to its offspring depends on whether or not the mutation is somatic or gametic. Mutations in somatic cells, such as cancer, aren't inheritable. A woman with breast cancer won't have a child who has breast cancer from birth, since, of course, a child does not inherit breast cells from her mother. Gametic mutations in egg or sperm cells, however, are heritable. For example, Down syndrome is a gametic mutation, and parents with Down syndrome are very likely to have children with Down syndrome as well. And that is a cause and potential impact of a mutation on an individual and its offspring. The end.